Marty Berkstead, AIChE, here with Stephanie Lamb from North Carolina State University. And Stephanie's here is going to talk to us about her presentation, A Mechanism of Foam Stabilization by Biopolymers. What's it all about, Stephanie?
Hi, so basically the idea behind this work is that we're studying how biopolymers can stabilize foams. So why do we want to use biopolymers for foam stabilization? Well, we're interested in studying these kinds of systems because biopolymers like cellulose and lignin, they're renewable materials, they're non-toxic to the environment, and they're environmentally friendly, so they have a lot of potential for making sustainable products. And so we've made particle stabilized foams from lignin particles as well as from cellulose particles in our lab. And particle stabilized foams from lignin particles can potentially be used for water remediation processes. And so if you have heavy metals in water, then you can potentially use this foam to absorb that contaminant. And with the hydrophobically modified cellulose, we've made foams that have functional properties, such as foams which can have selective colorization, and also foams which can respond to a magnetic field. And especially this magnetically responsive foam, it can also have potential applications in water remediation processes. So say you have a decon foam. Normally when you apply a foam for decontamination processes, someone actually has to come and vacuum up the foam or scoop up the foam. But if you have a foam which can respond to a magnetic field, then when you apply it to a chemical contaminant, then you can just spray the magnetic foam onto this contaminant, and after it neutralizes the chemical or biological contaminant, you can just collect the foam into a very specific area using a magnetic field.
So how do you magnify a foam? You just shave a magnet into it?
No, actually, so basically what we do is we incorporate iron particles, which are magnetically responsive, into the matrix of our hydrophobically modified cellulose, which is the foam stabilizer. And this causes the foam to become magnetically responsive.
Okay.
Yeah, and basically the work that's presented here is centered around exploring how biopolymers such as lignin and cellulose can stabilize foams, because this actually is not a very well researched area. So to do this, one of the things we did was we used an apparatus known as a thin film pressure balance, which basically simulates this area between two bubbles in a foam. So in the thin film pressure balance, you would draw a thin liquid film containing either the biopolymer, which is your stabilizer, or a combination of the biopolymer and the particle. And it's bound by two air phases, so it can simulate in 2D exactly this area in a foam system. And by doing this, we were able to better understand how the hydrophobically modified cellulose, which we were using to make the magnetically responsive foam system, could impart stability onto these foams. And basically what we found was that the first adsorbed layer in these foams is a molecular layer of biopolymer, and the layers adsorbed subsequent to this layer are then layers of the cellulose particles. And the more particles we add, the more stable the foam, because eventually when the liquid drains out of the foam through aging processes, which are normal to foam systems, or normal in foam systems, the cellulose layer will then compress and basically gelify, and therefore sterically stabilizing these types of foam systems. So basically that's what we learned so far, and we plan to tie in these 2D experiments a little better with the 3D foam system by just exploring rheological properties and how bulk rheology of these particles in suspension can affect foam stability. And also we would like to learn more about the biopolymer molecular layers that are adsorbed at the air-liquid interface. And to do this, theoretically it would be best to use a technique known as neutron reflectometry, but we don't really have access to that, so I guess we're not really sure how we're going to study this layer yet. So yeah, that's about it. But
thank you very much, Stephanie.
Yeah, no problem.
All right.